Hey everybody, we are going to continue talking about evolution and we're going to specifically look at speciation. So how new species are formed or how they occur. So we've got two general types of speciation that we're going to look at. We're going to look at allopatric and sympatric. And so both of these types of speciations work through a block of gene flow between two populations. It's just the type of block is a little bit different. So allopatric speciation, allopatric means other homeland, and so in this case you have an ancestral population that's going to be split by some geographical feature. So the size of the geographical feature could be very large or it could be really small because it's going to depend on what kind of species that you're looking at. So an example would be pupfish populations in Death Valley. And this generally happens when a species range shrinks for some reason. So here we have the fish within a lake, and you can see that the lake is kind of oddly shaped, and so what happens here with allopatric speciation is that the geographic feature that would change would be the lake, and so the center of the lake is dried up. It separates the two populations so they become two different species. With sympatric speciation, you can see the lake is still continuous, but we have a second species arise. So there are certain conditions that favor allopatric speciation. Founder's effect with the peripheral isolate is one. Genetic drift gives the isolate population variation as compared to the original population. And then you also have selection pressure on the isolated population that differs from the parent population. And so this is seen a lot on like the edges of habitats. Like the environment is different on the edges of a habitat. So like if you're looking at a forest, the edge of the forest is going to have slightly different environment. Like you get more sun towards the edge, you might be exposed to more predators. And so essentially you're just getting slightly different selection pressures. And the result here is that the gene pool of the isolate population changes from the parent population, and that allows the new species to form. All right, so changes in the gene pool or changes in allele frequency in this new isolated population is going to kind of be what causes that new species to form. Now with sympatric speciation, sympatric means the same homeland, and this is when you have a new species that arises within the same range of the parent population. And actually what's interesting about sympatric speciation is that it can occur very, very quickly, like even within a single generation. Here you can see, this is the sympatric example. You have a population of trees, and now you have a new population of trees that's still within the same location. There's no geographical feature that's separating them. This is really common in plants. So with plants, polyploids can cause new species because the change in chromosome number creates a postzygotic barrier. So I think we talked about this a little bit before, that plants can easily form polyploids, whereas animals don't tend to be able to do so. And those polyploids, because of the change in chromosome number, it creates a postzygotic barrier. So the plants that have that new polyploid number can no longer mate with the original plants that do not have that number. So polyploid types, you've got autopolyploid. This is when a species doubles its chromosome number from 2N to 4N, and it can then form a new species. And then you have allopolyploid, and this is formed as a polyploid hybrid between two species. So here you're taking a look at autopolyploid. So we have the karyotype of the parent species here, and so you can see that you've got 2N equals 6, so we've got 6 chromosomes, or 3 pairs of chromosomes. With some type of meiotic error, you now have gametes that don't have half the number of chromosomes, so they still have that 2N number, so each gamete has 6 chromosomes. Now if this plant were to self-fertilize and these two gametes come together, you now have a 4N tetraploid. So now the chromosome number in this individual is 12 instead of 6, and because this plant can continue to self-fertilize, essentially it could start a new species with a brand new chromosome number. With allopolyploid, it's a little bit more complex. And here you have a hybrid forming. So you've got species A, which is 2N equals 4. Here you've got species B with 2N equals 6. So again, you can have a mitotic error, and so now you have the chromosome number is not reduced from 2n to n. So again, we've got the 4 here. Here we have a normal gamete. These two can form a hybrid with 7 chromosomes. And now we have unreduced gamete with 7 chromosomes right here. Now if this unreduced gamete with 7 chromosomes were to fuse with a normal gamete from this original species, now you have a 2n equals 10, so we're back to an even number of chromosomes, and now this would be a viable fertile hybrid because it's got an even number of chromosomes. So you can see the autopolyploid is much faster, much simpler. The allopolyploid becomes a little bit more complicated. Adaptive radiation is when you have a rapid emergence of several species from a common ancestor. A lot of times this is happening through allopatric speciation. This is really common in islands or mountaintop populations or any environment that is considered empty. So if there were some natural disaster that kind of cleans an area out, it might be empty, and so now new individuals and new populations can migrate in, and this is really good for adaptive radiation. So adaptive radiation works when resources are temporarily infinite, meaning there's a lot of resources available for individuals and populations to use within an area. Because there are so many resources, most of the offspring are going to survive, and so you end up with very little natural selection, and that allows the gene pool to become just very, very diverse. And then later on, when the environment saturates or the resources are no longer infinite, then natural selection will start to resume. It'll kind of kick back up. And now new species are going to form rapidly if the isolation mechanisms work, meaning that if you have this huge population that's really diverse because they had infinite resources for a period of time, but now you run out of resources or you start to run out of resources, now natural selection is going to sort of pick the winners or the fittest in the population. And so maybe you've got individuals over here that compete really well and individuals over here that compete really well, and they are different enough from each other that then after natural selection sort of starts to select these two individuals, they may not be able to mate anymore because of, it could be the prezygotic barriers that we talked about, or it could even be the postzygotic isolation barriers. Some examples were the finches in the Galapagos Islands, African violets. So a quick question. So if isolation fails and populations come in contact with each other again, what do you think happens? So if you have these, individuals that have been separate, or these populations that have been separate for a long time, but they come back in contact to each other and there are not successful like prezygotic or postzygotic barriers, what can happen is you have an area of that we call a hybrid zone. And this is a region where members of different species meet and can mate and can produce hybrid offspring. All right, so here we have an example. So we have 5 individuals within a population that are shown. You can see the gene flow as the arrows going back and forth. What happens then, these populations over time are going to sort of separate as these guys go this way and this population goes this way. You can see now that the individuals inside are changing colors just to show that it's a different population, perhaps a new species now. But we have this area right here that we call the hybrid zone, and this is an area of overlap. And so individuals from this population and from these populations can meet in the hybrid zone, they can mate, and they can produce fertile offspring. So we can have 3 possible outcomes. We can have reinforcement, which means that the barriers will hold and the hybrids will eventually disappear. So maybe they suffer from hybrid breakdown and they don't compete as well as the original 2 species. You can have fusion, and this is where the barriers are going to essentially give way, and the species will fuse together because they're not different enough. So those isolation barriers don't work. Or you could have stability. And that means that the barriers are constant. However, because you have that area of sort of overlap, although the barriers are constant, you still are going to be producing hybrids. Okay, so again, you have reinforcement, fusion, or stability. So your lecture question for today, so don't forget to send me a message via the class website is, how long do you think it takes for a new species to form? So we talked about 2 different types of speciation. Let me know what you think about allopatric speciation and sympatric speciation. And I hope you guys have a good one. Thanks.